Thank you. The dedicated team out here fighting the beach. I get it. You're making sacrifices. I appreciate it. So Deddy's always a hard talk to fill. He's got big shoes. He gives great talks on cancer. Unfortunately, he can't make it to the Israel stuff that's going on. But I figured what I'd do is I'd fill in on discussing what's been very hotly debated throughout this entire conference. Which is what is going on with the differential regulation that we have, and what fingerprints does this leave in the sequencing data and in the microbial data that we have. And so I asked AI to sum up this and make me a little picture, and it came up with this circular firing squad on top of a Titanic filled with cannabis. And it is it's meant to symbolize the fact that we do seem to have two markets that are chasing the same plant but fighting. And we also have lots of consternation right now over laboratory shopping, which is a valid concern, and we'll show you why. But the most egregious form of laboratory shopping is not ever doing any testing, which is what we have now legalized throughout the state or throughout the country through the Farm Bill. And so this study will go through some of the tools we've built to monitor safety in a different way. And it will also go through some of the economics that are behind this, because if you don't pay attention to the economics, you're probably going to be digging in the wrong place for a while. And the economics, I was shocked to find this year, just reading a couple of these reports, have really changed quite a bit. For those who don't know medicinal genomics, I think you probably know us by now, since you're here. But we do a lot of different things in the space, in that we make microbial testing tools, usually PCR-based with some of the robotics you saw out there. We do a lot of plant and virus detection. These aren't necessarily regulated tests. These could get used in either the hemp or the cannabis industry. And we build automation tools for a lot of the labs that are out there doing safety testing. There's also some plant genetics work that we do. We do a lot of sequencing of cannabis genomes. I want to share what we've learned from that. We have over 2,200 cannabis genomes now sequenced, so that it's given us a very unique picture on the microbiome. And this also helps us understand how to grow cannabis in certain environments, because we are finding, if you guys saw Zamir Punja's talk this morning, we've all been trying to scrub the environments in our grows to make sure we don't have any of these pathogens getting in, but what they're finding now is that a lot of these things, we think the entry is coming from the substrates we're growing in. The very cocoa and the rock wool that we're using seems to be seeding the plants with these contaminants, and they're trickling all the way through to the point where they can be failing people's total use of mold tests and what have you. So the hemp industry, you'll probably be shocked to find out, is now bigger than the cannabis industry, and this happened in 2 years. It's a really rapid growth of grows shifting and running away from the cannabis industry to become hemp companies, because there are several advantages to doing this, which we'll lay out. These are based on Whitney economics. They have this estimated anywhere between 28 billion to 80 billion, depending on how you measure the additional multipliers on the economy. But we have the cannabis industry from his same estimates is around 34 to 35 billion. So we're getting a hemp market now that is actually larger than what we have. In the last few years, if you look at the growth of the cannabis industries, many of the MSOs have all sort of stalled in their sales. They're flat for 2 or 3 years. And if you look in the graph over on sorry, I'm shooting you guys. That's the cannabis industry. There's the hemp industry in the same time frame. So it has eclipsed the industry in a really short period of time. So there's clearly demand for things going that direction. Now, what's driving some of this demand? You can look to Google and do a quick search on THCA flower, and you'll see a rapid rise in the interest of that term ever since the Farm Bill. Probably not surprising. And then there is this curious increase of smoke shops everywhere. So these are smoke shops licenses. This individual on Twitter pulled them all down from, I think this is Florida, Georgia, and Texas, and they are going exponential in growth of smoke shop licenses in many of the red states that have not legalized recreational cannabis. And of course, we've seen there's been some lobbying from the hemp industry to keep cannabis recreational legalization in Florida. They actually were lobbying against that. So these two industries have now been pitted against each other, and hence the circular firing squad I was referring to. Now, this recently came out from some of our attendees here. I think Josh Schweider's somewhere here. You may have met him. He's doing work at InfantCal, and Tiffany DeVitt, everyone knows from Martin Lee and Project CBD. They've been doing great work. And I think the important thing to say here is that the hemp industry is one of the inspirations for us actually having this conference, because they were the industry that supplied kids with CBD products many years ago. You might know of Charlotte's Web or Miriam's Hope. We've done work with even our own family members. We were buying these products from healer.com and others to get access to high quality tested CBD products. But now what they're finding in all of these smoke shops is so far from what these companies represent, yet they're kind of getting labeled as the same industry, which is unfortunate. And I think some of the bills that are getting shoved around in Texas and in California, they're talking about banning all of those people who have been here for decades saving these children. Because what we have now are a high rate of synthetic cannabinoids being sold in gas stations and in smoke shops with very limited age gating. They're selling it over state lines. Now, the economics behind this is obvious, right? They don't have 280E to contend with. They can sell over state lines. They have limited age gating. So the taxes are like a phenomenal incentive for these people to move there. And they also do not have any safety testing. So it's easy to slip in ISO Delta 8 or THCP or HHC into these products, and you can call them a hemp product, and the customer probably won't know the difference until they've had a 500 milligram Delta 8 gummy, which killed a kid in Georgia very recently. 24-year-old actually was reported having died. Now, there's a curious case on that death in that they didn't claim it was the cannabis. He had tachycardia and a heart condition, but I think anyone knows if you have a 500 milligram Delta 8 gummy, your heart's going to probably race a little bit, and it very likely aggravated his condition. There's a case like this in Rhode Island as well that was a similar death coming from one of these type of gas station products. So encourage you to read this study. The Great Hemp Hoax was shocking. If you read this, you'll see how many contaminants are actually in this field. And what it looks to be, from our perspective in the testing facilities and in the space, is that the products that are failing in the regulated space are just getting sold out the back door as THC hemp and going to all these other red states that don't have access. So we have a couple different data sources that we use to track this down. A really important data set came out from MCR Laboratories. I'm sure people have seen Yasha Khan's work on this. This was all of the laboratory testing data from 14 different states that he FOIA'd. I think the list has grown longer because he's not stopped. He's a dog and a bone with this. And we downloaded it at a certain imaging point where we got 14 of the states, and then just to do a quality check on his own work and make sure that there was no conflicts there, we separately FOIA'd the state of Massachusetts for the same data and lined them up, and they were in fact the same. The state tried to hide some of the metric codes and made them different for him as they did for us, but we were able to resolve all that. But the actual testing date and quantities and the pass rates all lined up. So we built this web testing portal so people can go into the Medicinal Genomics website, go to the lab testing portal, and you can then look at the pass fail rates for all the different tests that are being done in the microbial space. I think there's some THC data in there as well. I think we're working at still getting some of the pesticide data in there. But the reason we wanted this is that we've heard all this accusation about lab shopping. The only way to solve this is with transparent data. Put all the regulatory data public and let all the internet nerds go crazy with it, and we'll start to find trends, and everyone will sharpen the knife and hopefully get better. It's very hard to have all of these decentralized labs when no one can actually see what their performances are. I think once this data comes live, it becomes much easier for labs to recognize that, okay, we're a little bit over, we're a little bit under, maybe we need to tune our methods to be more in line with the PTs. So I think it's a very helpful, transparent system. So here's a couple examples that I think has helped us in Massachusetts to understand why is there such a pressure for lab shopping. Again, this is the regulated industry where you have to test. So just keep in mind that when you're in the hemp industry, none of this has to be done. So this is looking at total yeast and mold. The total yeast and mold test is probably responsible for the most fails in the cannabis industry, sometimes upwards to 10 to 15% failure rates. And you'll see one of the labs on the top has a very sort of normal distribution of data, if you will, but it's broader than the other two labs below. What you're looking at those dot plots are this is the total yeast and mold colony count, and this is over time, and each dot's a test. And this little red line is the 10,000 CFU threshold, that if you're over that line, you fail, and if you're under it, you pass. So this is an interesting laboratory that looks as if their yeast and mold have learned to never cross that threshold. So that's an oddity. That's something that's bizarre. This is a little bit more normal data, and this is perhaps has higher failure rate. This is the highest failure rate lab in the state, and you can kind of see them in this bar graph here. Now, there's one I couldn't even chart because there's one lab in Massachusetts that's never, ever failed a sample for total yeast and mold, which is another extreme outlier. So this does beg the question, how do we know who's right in this circumstance? I mean, artificial data like this looks like there's an artifact, and there is a certain platform that's being used here that perhaps is getting saturated at that level. But one way you can get to some answers to this is you can start comparing these state to state and see how they behave in different states. Some of the same labs exist in other states, and sometimes they exist in different states with different regulations. And so this is another angle on this. But an important topic I think everyone needs to know about when it comes to total use and mold testing, we actually don't have a truth set. We don't know who's right because we don't have standards. There's a standard that floats around the industry that has 2 organisms in it, but we're honestly trying to measure millions here. And so a 2-organism standard is really not a standard, it's just something that leads to lab shopping in our opinion. So we've done a lot of work on this, sequencing many of these things, and we can tell you that many of the culture-based systems they tend to overcall because some bacteria ends up growing on these plates. They can't perfectly get rid of all the bacteria, so they tend to have a higher fail rate if you're using culture systems. The flip side is if you're using DNA systems, if you irradiate the samples, sometimes the DNA systems still fail them because there's still some residual DNA around, so they can have a higher fail rate for samples that have been irradiated versus culture. It's very easy to counterfeit a culture system by zapping it or microwaving it. So there are pros and cons, and some states use irradiation and some states don't, so you have to be aware of what state you're looking at for this data. Here's an example we did many years ago where we actually took Pseudomonas aeruginosa, which is a bacteria, and we took that bought it from ATCC, a reference standard, and put it into some of the bioMérieux Tempo platforms, and they would trigger a false positive total use and mold hit. So we know this is happening. We've even seen this on 3M's Petri films. Their own product information will tell you that their inclusion-exclusion criteria, that like 30% of the material that comes through on their plates is bacteria. All right, so we don't have a perfect truth set when everyone says you got to make the molecular systems do the exact same thing as the culture-based systems. The only way we do that is if we make mistakes on the DNA front. Because on the DNA front, we don't confuse these things. It's ITS sequencing or 16S sequencing. It's very ironclad differentiation between bacteria and yeast and mold. So you can take one of the states in Massachusetts, that state that was like in the middle, where we don't really know who's right or wrong. You look at that same lab in Maryland that has a 100,000 K limit, and something different emerges, which is that they have the highest failure rate in Maryland, but they also have the highest volume, which contradicts the lab shopping thing. What was going on in Massachusetts is the lab that would never actually pass the 10,000 CFU threshold was getting all the volume. That makes sense, but that's because the fail rate there was 15%. So what this is telling us is that when the total use and mold tests are in place at low thresholds and you're getting 10 to 15% failure rates, you create economic incentives for lab shopping. THC is also a driver in this, so that's probably playing a role here. When you go into Maryland, who has a 100,000 CFU limit on total yeast and mold, we don't see the lab shopping artifacts on the yeast and mold test because the people with the highest failure rate have all the business. The graph over here is the volume of tests, and this is just normalized. You can get a sense of the percentage. So you can see they have a higher percentage failure rate, but they also have accumulated all the business. So many states have moved to 100,000 or 100,000 CFUs for total use and mold because they're not finding this test to be very have any clinical meaning. And I'm going to touch on that. That's, I think, a very important point as we try to refine these tests. Now, the reason I'm bringing all of this forward to you for the complications we have in the cannabis space is that if we continue to drive with some of these tests that we don't think have any clinical literature supporting them, we're just going to drive people into the hemp market because they don't want to deal with 15%. I mean, lab shopping is one way to deal with this, but the ultimate way to lab shop is to just put on the hemp hat and be done with this. You get better taxes. And so this brings us to asking, what are the most clinically productive tests that we can be running in the cannabis industry? If you look at all of these total count tests that do not speciate the organism, they're looking at total coliforms, total yeast and mold, total coliform, and total biotolerant gram-negative. Those are all the tests you see over here. So this is the failure rate across 14 different states on those total count tests. And then you start looking at the species-specific tests, and they're orders of magnitude lower. So this has been something we've been singing for a while, that we would prefer to be using these types of tests because we can make standards for these tests. As a specific organism, you can get it from a biobank, and you can prove that you're hitting it or not. We can't make standards for any of these tests because they encompass thousands, if not millions, of organisms. We could probably do that with the DNA. We can synthesize that on a chip and make a million different pieces of DNA and prove that, but we can't do that with living organisms for the plating people, right? They'd have to actually figure out how to culture those, and 95% of them they don't know how to culture. So these are what I would call tarot cards. They're standardless tests that have been put on the industry because of a USP article that came out many years ago suggesting these might be good qualitative assessments for whether your grow has got too high or too low, but they shouldn't be used to fail a particular sample. They are, at this point, being used for that, and it's creating a lot of this mayhem. So we're also able to pull out of the California data from Yasha and look at what are the pesticide failures compared to the heavy metals, compared to the microbials, because one of the concerns that's been raised in the industry is that if you have really stringent total yeast and mold or total count tests, you are going to drive really high pesticide use. That's not holding up in the California data, but you have to note that California got rid of total yeast and mold testing and went in the direction of species-specific Aspergillus, E. coli, and Salmonella testing. And you can see that their actual microbial failure rate has been dropping and is going below 1% now for all tests. And you can see that the pesticides are dropping with it. So we're not seeing an increased use of pesticides as they're putting in these species-specific tests. Okay, so the other thing we can do, now that we have all of the testing data of the labs and how they're performing and which tests, we can then intersect that with a very large genomics database that we've built. We have now 2,200 genomes sequenced from cannabis, and about half of those genomes we sequence with a method known as whole genome sequencing. And when you do whole genome sequencing, the whole microbiome comes through with it as well. So you can start classifying what is the microbiome on these plants, and does that actually line up with what we're seeing the testing lab screen? And can that inform what we should be testing for as opposed to just guessing with these total tests? So this results in some of these corona plots you probably saw a few other speakers mention. So there's about 1,244 genomes that are public now in Cannapedia, and with them come these little corona plots. I shouldn't say corona, it's corona, no virus involved. And these things give you a breakdown of what all of what the microbiome looks like in these. Interestingly enough, there's a good portion of these things that are now segregated on 4 different tissue types. So we have a bunch that are from roots, a bunch that are from stems, a bunch from leaves, and a bunch from flowers. So we can also begin to answer questions of, are these endophytes that are coming through the roots, or are they coming in sprinkling from the air? In the case of Aspergillus, what you're seeing here is a waterfall plot of the frequency of Aspergillus nigrotarius, fumigatus, and flavus, which are the 4 that we test for, amongst the background of another 50 Aspergillus genomes that we can pick up in the sequencing of these plants. So there are lots of different Aspergillus organisms in there, but we're only going after these particular 4 species. So you may hear from a lot of people that Aspergillus is ubiquitous. What they're talking about is all 400 species that exist in the world, but we're only testing for 4 of them, all right? So that's the reason why the fail rates are so low in some of these testing labs. So this is an example of looking at the tropism of Aspergillus in the plant. You can look at these 4 different Aspergillus species and see that in the roots, we're looking at 10 to 1,000 reads per million is the bar graph that you're seeing down below here. And in the stem, it's very similar. And you get up to leaf, it's more like in 100 to 10 to 100 range. And when you get to the leaf, it's like the flower, it's 1 to 10. So going up from the roots, the stem, the leaves, the flower, you can see it drops about 2 orders of magnitude in the frequency. So it's coming in from the roots, it's an endophyte, and that was actually very prescient because what we saw from Zamir is he was showing that it's not only in the roots, it's in the actual cocoa fiber and the rock wool. So that is the source of a lot of this, is that people are starting their plants off in substrate that's contaminated, and it makes its way all the way through to a failing sample. So screening those things is very important. The other thing we can do is ask ourselves, okay, what about these other, you showed us a total yeast and mold test that was counterfeited by a Pseudomonas. It was positive for Pseudomonas, which is a bacteria. How frequently is that found in the database? Well, it's right up there, the top 50 of the bacteria that we ever find in cannabis. So it's very likely that this is causing a lot of total yeast and mold failure in the environment. Now, this does need to be double checked. One thing that Zamir is doing is he's repeating some of this work after curing, because some of these bacteria will sporulate and survive curing and some won't. And we don't have that data yet, but it is certainly present there in all these different tissues. What he did point out when we actually get to the fungi is that there are different beneficial microbes that people like to use. Now, you can get yourself in trouble using these beneficial microbes if you're in a test that uses total yeast and mold, or a state that uses total yeast and mold testing, because it becomes a de facto ban on using beneficial microbes. that's somewhat true depending on the microbe. So what he showed is that yes, trichoderma is a wonderful beneficial microbe you can use to push out aspergillus. However, you don't want to use penicillium. Penicillium chrysogenum is often used as well because that one, when you cure it, doesn't die and it triggers the test. Trichoderma seems to die during curing and it doesn't trigger the test. So the choice of your beneficial bacteria and fungi are really important if you want to evade some of these, what we call somewhat counterproductive testing that's going on. And you can see some of these similar beneficials in the bacteria side. There's some bacillus species over here, and we're still doing some work to see like how do those survive going through the curing process. But the reason I raise this is that when you do these total yeast and mold tests or total aerobic count tests, you are putting on a lot of pressure on a grower to not be able to use the microbiome of the plant, which means they're going to have to start using fungicides and pesticides, and that's a dangerous direction to push them because we only test for 66 of those in some states, so you get outside of those and you can use them, and we don't have a cumulative metric on those. So you can use 10 pesticides that are all under the limit, but cumulatively they're not penalized, right? So that's what we're finding people do, is you look at a lot of the pesticide fails, it's not a single pesticide, it's like 5 or 6 they're trying to put on at very low levels so they pass the testing, but cumulatively they're damaging enough to stop the problem. That gets to be a little bit more, I think, complicated from an epidemiology standpoint to track. We have lists of archaea, and interesting DNA viruses come through with this. We don't get the RNA viruses. 80% of the pathogenic viruses are RNA, so this is interesting to look at what's going on with the DNA viruses, but I'd be very surprised if those are pathogenic. Okay, so now let's dig into some of the information that's on our poster out there. To use this pipeline, we then set out and got 30 samples from the hemp industry, THC hemp, brought them in, sequenced all their genomes, and ran some microbiome analysis on them, and then compared them to 15 samples we recently pulled out of the Massachusetts dispensary market. Now, the Massachusetts dispensary market is interesting. As I mentioned, they're using these total yeast and mold tests, which we don't think are very productive, and they're not using the species-specific tests on aspergillus. And so you'll see that actually has a consequence in Massachusetts. 4 of the samples we ordered twice from the same vendor and repeated this just to see if they were the same, and you'll find out they weren't. They were all different genetically. First thing you do is you check, like, okay, these got through the dispensary. Now, keep in mind, when people test these at a lab, there's then a packaging step and a shelf life that we don't understand. And so it's not uncommon for people to do secret shopping and get different results because it's sitting on a shelf for a period of time, and we don't know how these things replicate over time. And likewise, the testing that's done on a secret shopper program is often a 1 gram sample when the labs are forced to do large homogenizations and test much larger volumes, and so there's a Poisson sampling game going on that doesn't always line up. These were pretty close, actually. In this case, this is when they say in the microbial world, if you're under an order of magnitude and you're in the range, it's like horseshoes and hand grenades. They expect you to get close. They don't expect you to get the exact same number. So these technically were passing for TAC, but they were a little bit over for total yeast and mold. That's not too surprising for us. There were only 2 samples like this out of the 16 that had discrepant results in the Massachusetts dispensary system. So the other 14 were clean and passed just as they would expect. But what was interesting is these 2 samples also had Aspergillus fumigatus in all of them because we don't test for that in Massachusetts. So that's giving us a 2 out of 16 that were positive for Aspergillus. Now, you get into the hemp side of things, and this thing goes way up to 56% positivity for Aspergillus. All right, so it's clear. Now, what is that? How does that compare to like tested states that are used to testing for Aspergillus? Like you look at Nevada and you look at Michigan, who've been doing this the longest, and their Aspergillus failure rates are like 3%. All right, so you go to a state that doesn't test for it but has cleanliness in mind because they have to pass other yeast and mold tests. The number's like 12%, and when you go to the entire hemp market, granted N of 30 sample size here, it's like 56%. So the Aspergillus seems to be, if it fails in a state, probably ends up in this marketplace. All right, so there's another thing you can do with this data, is you can take all of the reads that come out of a cannabis genome that don't map to the cannabis genome, put them aside and assemble them into microbiomes. And when you do that assembly, you can start to build larger contigs of all of these fungi and bacteria, and you can run them through a program that starts to count the virulence factor genes that are there, the toxin genes that are there, or the antibiotic resistance genes that are there. And these are just charts that are measuring those 3 things, and what's interesting is the samples that are in red here are from the hemp industry, and they have a lot more of these antibiotic resistance and toxin genes. Zamir Punja, who knows exactly what he's doing, are the cleanest samples of all, no surprise there, and the Massachusetts dispensary in 2025 that isn't necessarily screening for Aspergillus, which does have toxin genes in it, are somewhere in between. All right, so this is another way to try to quantitate the microbial burden as to how it may be a risk to humans, because we're not very fond of this total yeast and mold thing that doesn't look at pathogenicity or virulence factors. All right, so how did they do on the chemotyping? So this work was actually done by Cannabis Analytics. They took these samples and they ran them through THC testing, and you can see all of these things are 15% or higher on THCA, which everyone here knows once you heat that it's THC. So these are not hemp samples by any stretch of the imagination. One sample was below the 0.3 limit on delta 9, but another 4 samples were CBD plants. They were hemp plants by genetics, I'll show you that, but they happened to have a delta 8 isomer on them. And now that delta 8 isomer is important because Mark showed us a really interesting presentation that seems to have different receptor binding activity than delta 9. All right, so they're spraying these with compounds that they could be a very interesting therapeutic compound, right? It needs to be studied, but it shouldn't be studied on gas stations on your kids, right? So I don't want to like belittle this compound that you found, but there's a better way to figure out how this is acting. But it's very curious, I think, your work showing that this may not be CB1 activity. This may be another receptor that it's acting on to create a high. These are the plots, the heat maps that we generate to look at the genetics, so we can tell whether something's hemp or cannabis by the presence of the THC synthase gene, or 2 mutations in it that knock it out. There's the way to do this. So those 4 samples that had delta 8 on them were in fact hemp samples genetically. They did not have a THC synthase gene. The only way that THC got on there was someone acid catalyzed CBD and sprayed it on. And that was not labeled on the product. It was just called hemp. They went through pesticides. 9 of the samples failed with 7 different pesticides. That's actually pretty good for the hemp industry. I've seen other studies from Josh and other people that this is I wasn't expecting this. I was expecting this to be a much bigger train wreck. And there were 3 samples that had some heavy metals over the limit as well. Now the genetics became very interesting because we started to look at how related were these. These were from 6 different vendors, 5 samples from 6 different vendors. And we started to compare them, and it looks like they're all using very similar genetics, but they're calling them all different things to try to obfuscate the chain of custody here. So all of these particular strains sold by different vendors were clonal on this table over here. They're identical, but they're sold by a different vendor with a different label. And a lot of them are siblings too. So it could come down to just a couple growers that are feeding all of these different distros with these genetics. Of course, when we reordered the same 4 all over again 4 weeks later, none of them were the same. So their manufacturing process is to, when they get an order, is to fill any can they have available with any weed that's present there. There is no genetic fidelity in the system. So we have some takeaways from this, which we put an article in with the Society of Cannabis Clinicians very recently because this has become a hot topic. Is there is a group of people running around saying we should get rid of aspergillus testing, and we're saying hold on, wait a minute. There's good reason to change what we're doing in the cannabis testing space, particularly with these total tests, but I think they're digging in the wrong place here. The tests that are really causing lab shopping that we can't defend clinically are the total count tests. The aspergillus test, that has the highest stack of clinical literature of harm with aspergillosis. It's the deepest body burden that we have in cannabis is on people getting sick with that thing, and it has a much lower fail rate. Now, a lot of this stemmed out of Oregon, because when Oregon booted this up, all the labs weren't calibrated yet, and some of the labs had like 60% failure rate. I would protest that too. That's way too high of a fail rate, and it did not match what we were seeing as 3% failure rates in other states. So this whole sort of tempest in a teapot occurred because we did not have transparent data. The people in Oregon did not know what the fail rates were in Nevada to know that, oh my God, the lab's having 60% failure rate. There's something wrong in the lab. It's not the cannabis. They're not calibrated. They have not calibrated their tests. So to follow up on this, we really need to have better standards on these things. And I see my time is ticking here, so I'll go quickly through these. We have done some sequencing to demonstrate that when you do total yeast and mold testing and then you go about and sequence all those colonies, depending on the media you use, you get very different results. And there's lots of bacteria that come through depending on which plating source that you use. On top of this, we see a lot of these bacillus things when we did the sequencing, and those bacillus organisms are probably beneficial and you're getting penalized for using them. And of course, if the language of a CFU isn't very definitive, if you move to different plating systems, you're going to get different counts. So they're asking all the PCR systems to match a standard that we really don't have any good standard for. So why does this matter? What matters is that in order for you to have clinical causality on something like this, you need to perform Koch's postulates. You need to actually identify and isolate the organism and then put it into a new host and demonstrate clinical harm. We cannot do that with a total test because you don't know what you're measuring. So we never can fulfill Koch's postulates, and that's why if you go searching through the literature for clinical harms of 10,000 CFUs versus 11,000 CFUs, you can't find anything because no one will get that through peer review. It's just, you have to perform this. So we have tests in place that can't satisfy Koch's postulates. If you can't satisfy Koch's postulates, you can't satisfy Bradford Hill's tenets of causality because you need to be able to satisfy the specificity requirement. So these tests will never be causally linked to any harm in humans, yet we are penalizing the cannabis industry so hard with these that they're all running and going to hemp. And so this is why we have been very vocal about we need to change this, and we need to move to species-specific testing where there is clinical documentation at the CDC for E. coli, Salmonella, and Aspergillus, and how much actual medical harm that these things cause, so that if we do end up in court with a grow saying you're failing 15% of our business on a test that is arbitrary and capricious, there will be literature to say this is the harm that will be induced if you relax this. We don't have that literature for the total tests. So, I don't have time for that. We'll just, I think I've covered why we think the species specific is important, but I really want to drive home that it's important for lab shopping not to occur. We need standards, and we do not have standards for the total tests. So we should abandon them or put them to a point where they're just indicator tests, where they're tests that can inform a grow about the bioburden they have, but not be used to penalize them financially. When that's in place, we can actually get standards for all the other microbes that we're targeting, and we can get literature that supports whether they're risky or not risky. And this will, I think, help advance the industry because as we discover new microbes, we saw a lot of discussions about fusarium. That's probably something that should be on the list because it can cause fusariosis and it makes mycotoxins like DON. But we're over here wasting a lot of our energy doing these total count tests such that no one can really focus on things that are prevalent in the plant and in fact have some clinical risk. So that's my story about the circling firing squad in the cannabis industry, and hopefully we can all get together and figure out how to get the regulations in the cannabis industry to be lighter, have banking, and the regulations in the hemp industry to be cleaner and a little bit more responsible as to what's on their label. Thank you.
Hello?
Yeah. Fascinating, Kevin. So I'm reminded of a comment that one of my DuPont colleagues used to say, it's a 3-ring circus and they keep moving the rings.
Yes, that's good.
So now hemp folks have gotten more sophisticated, and rather than spraying with Delta 8, they're actually adding THCA isolate to hemp flower. So now when you test that, it's going to show THCA and not Delta 8, but it's not going to have the synthase, right?
Yes, this is true. We will see it from you're right. So Mark's point is that the genetics will tell the tale as to whether the plant was capable of making this or not. And that's very easily done with sequencing. And even some PCR tests we have can pick it up. So thank you.
Any other questions?
I think Sherman's got one back.
Thank you very much, Kevin. In your identification and detection of antibiotic resistance genes, did you detect any anti-fungal genes to like azoles, which are the primary medications to take care of fungal respiratory infections?
That's a very good point. That would be a nightmare if we found those, because that would imply we're spreading around genetics that would lead to antifungal resistance in patients, right? Yeah, I'll ask Steve about that to see if we can go looking for those. It's a very good point.
Thank you very much.
All right, thank you, Kevin.
All right.